everyone, this is Dr. Vishal Trivedi from Department of Biosciences and Bioengineering, IIT Guwahati. And what we were discussing about the central dogma of life or the central dogma of molecular biology. And in this context, so far what we have discussed about the DNA dependent DNA synthesis or the replications, and we have understood the many aspects of the replications and how the replication the steps what are present in the replication for the synthesis of the new DNA strand can be modified or can be performed under the in vitro reactions, so that you can be able to amplify any given DNA strands, and that is a very robust techniques which we have discussed in the previous lectures, and that technique is called as the polymerase chain reactions. Subsequent to that, we have also discussed about the DNA dependent RNA synthesis, and that event is also being catalyzed by the RNA polymerases, and that process is called as the transcriptions. So in the previous lecture, we discussed about the differences between the transcription in the prokaryote versus eukaryotes. We have discussed about the different contrasting features of the transcription in the prokaryotic versus eukaryotic cells, and how it is actually being different. So the transcription is different in three major aspects between the prokaryotic versus the eukaryotic cell. First is the site of transcription. The site of transcription in the case of prokaryote is within the cytosol, whereas in the eukaryotes it is the nucleus. And the second contrasting feature is that the transcription and the translation both are occurring simultaneously in the case of the prokaryotes, whereas it is occurring differently, like the transcription is occurring inside the nucleus, whereas the translation is occurring inside the cytosol. So that also gives regulatory events, and where you can actually be able to control the transcription and translation boards separately in the case of eukaryotes. The third is the transcription does not require the transcription factor in the case of the prokaryotes, and in the case of eukaryotes it requires the different types of transcription factors to recognize the promoter regions, and that's how the transcription is going to be more tightly controlled in the case of the eukaryotic cells. So in the previous lecture, we also discussed about the different events, the structure of the RNA polymerase in the case of prokaryotic cells, and we also have discussed about the different events like the initiation, elongation, and terminations. So in today's lecture, we are going to start discussing about the transcription in the eukaryotes, and then subsequent to that, we are also going to discuss about the post-transcriptional modifications. So let's start discussing about the transcription in the prokaryotes, in eukaryotes. So transcription in eukaryotes is different from the transcription in prokaryotes, right? It requires the entirely different machinery, right? So eukaryotic transcription is different from the prokaryotic transcriptions. The eukaryotic transcription requires the transcriptional binding factors, enhancers, and it also requires the RNA polymerase. Remember, when we were talking about the prokaryotic transcriptions, we said that the prokaryotic transcription only requires the RNA polymerase, and that RNA polymerase is sufficient enough to recognize the promoters. It also can unbind the DNA, and then it also can form first the closed complex and then the open complex to initiate the transcription, and then it also can enter into the elongation, and then eventually it can reach to the terminations. Whereas in the case of eukaryotic transcriptions, you require the transcriptional binding factors or the transcriptional factors, you require the enhancers, and then you also require the RNA polymerase. So what are the transcription factors? So transcription factors are the proteins which are essential for the transcription, but these are not part of the RNA polymerase. These factors are binding at the DNA template sequentially, and then the RNA polymerase binds and forms the initiation complex. The basal transcription factors create a structure at the promoter, so RNA polymerase can easily recognize to the promoter. In bacteria, single RNA polymerase can transcribe the all type of RNA, but in the eukaryotes, it requires the different types of RNA polymerase for the different types of RNA structures. So you know that there we are having the three different types of RNA molecules. We have the ribosomal RNA molecule, we have the messenger RNA molecule, and then we also have the transfer RNA, right? So, or tRNA molecule. So all these three different types of RNA molecules are transcribed with the help of the different types of RNA polymerase, whereas in the case of bacteria, all these three species of the RNA molecules are being transcribed by the single RNA polymerase. So that is also a very big difference between the eukaryotic and the prokaryotic transcriptions. So let's first discuss about the machinery. So the machinery in the case of the eukaryotic transcription is the RNA polymerase. So the RNA polymerase of the mitochondria and the chloroplast of the eukaryotic cells are similar to the bacteria, but all eukaryotic RNA polymerase are multi-subunit protein which contains three different multi-subunit proteins. It contains three different types of RNA polymerase which is responsible for the transcriptions. So you can have the three different types of RNA polymerase. You can have the RNA polymerase 1, you can have the RNA polymerase 2, you can have the RNA polymerase 3. The RNA polymerase 1 is required for the synthesis of the ribosomal RNA, and it is resistance for the amanitin, okay, and it is present inside the nuclei, okay. Then we have the RNA polymerase 2. RNA polymerase 2 is responsible for the synthesis of the messenger RNA, and it is very sensitive for the amanitin treatment, and it is present into the nucleoplasm, which means it is present inside the nucleus, right? And then we have the RNA polymerase 3. RNA polymerase 3 is synthesis of the transfer RNA, and it is less sensitive, and it is also present in the nucleoplasm. So you can see that it has the three different types of RNA polymerase. You have the RNA polymerase 1, 2 and 3. Their location, their sensitivity to the amanitin, and their role into the transcription is also very different in the case of eukaryotic transcriptions. Then apart from that, you also have the eukaryotic promoters. So eukaryotic promoters, each promoter contains some specific sequence which get recognized by the transcription factors. So remember that in the prokaryotic transcription you have the similar kind of transcriptional arrangement, like you have the -10 box, you have the -35 box, and you all this kind of Prinbo box, Tata box, and all this kind of thing. But that kind of arrangement is not present in the eukaryotic promoter. Eukaryotic promoter has the specific sequences which are being recognized by the transcription factors, and the eukaryotic promoter has a longer region than the prokaryotic promoter because it contains all those sequences which are important regarding to the initiations. It includes the core promoter element at which the RNA polymerase get attached and form the initiation complex, and also for the efficient transcription it requires the upstream promoter element, which are basically the GC rich region at which the transcription factors are going to bind. So transcriptional, the eukaryotic promoters are way bigger than the prokaryotic transcription, because the eukaryotic promoters are going to have the many regions, and these regions are going to be responsible for binding of the RNA polymerase, and on the other hand it also requires the binding of the different types of transcription factors, and that's why the transcription in the eukaryotes is going to be tightly regulated compared to the transcription in the prokaryotes. Then we have the transcriptional initiation. So transcriptional initiation by the RNA polymerase 2. So this is, we are discussing about the transcription of the messenger RNA. Okay. So we are going to have the different types of transcription factors. So eukaryotic messenger RNA transcription require the initiation complex, which consist of the general transcription factors and the mediators. So we have the many types of general transcription factors. So general transcription factor, we have the TF2D, and that TF2D is actually going to use to recognize the core promoter like the Tata box, or also going to recognize the core promoter like the non-Tata boxes, like other kind of sequences. So it can also have the two different types of proteins. You can have a Tata binding proteins, which is called as Tata binding proteins, or it can also have the TFEs, which is called as the Tata binding protein associated factors. So within the TF2D you can have the two different types of proteins. Then you can also have the transcription factor 2A. The transcription factor 2A, the function of the transcription factor 2A is that it stabilizes the tata binding proteins and the tata binding protein associated factors. Then it also requires the TF2B. TF2B is actually helping the RNA polymerase 2 and the TF2F recruitment, and also helps in the start site selections. So TF2B is very important because it is actually going to provide the docking site for the RNA polymerase, and it also provides the docking site for the TF2F, and both are actually going to help in terms of the selecting the transcription start site. Then we have the TF2F. TF2F is going to help the RNA polymerase in promoter binding. Then we have the TF2E. TF2E is going to help in the TF2H recruitment and the modulation of the TF2H helicases, ATPase and kinase activity. Then it also requires the TF2H. So TF2H is going to help in promoter melting with these helicase activity and the promoter clearance by the phosphorylations. So let's see how these transcription factors or the general transcription factor are going to participate into the transcription of the eukaryotes. So the transcriptional initiation by the RNA polymerase 2 is going to be performed by the not only one factor but by the many factors. So these transcription factors are sequentially going to bind the Tata box DNA to form a pre-initiation complex. So what will happen is that you have this is the promoter, right? So you have the Tata box and then you have the other promoter regions. So the first transcription factor which is going to come and bind is TF2D, and when the TF2D is bind, it's actually going to provide the docking site for the TF2A, and then from the TF2A it is actually going to bind the TF2B, and the TF2B is going to provide the binding site for the TF2F, and it's also going to allow the binding of the RNA polymerase, right? So that's RNA polymerase 2 is actually going to bind, and then its TF2F is going to provide the binding site for the TF2E, and the TF2E is eventually going to allow the binding of the TF2H, and the TF2H is actually going to allow the recruitment of the helicases, and that's how it is actually going to start unbinding the DNA, and that's how it's actually going to help the RNA polymerase to initiate the transcriptions. So at last, when the TF2H is going to bind, it phosphorylates the RNA polymerase 2 to initiate the transcription in the presence of ATP. So why this the phosphorylation is important? Because the RNA polymerase will go and bind to the DNA because it's going to be positively charged and the DNA is going to be negatively charged, and that's why when in the last step the TF2H is actually, when it binds, it actually going to have the intrinsic kinase activity, and that intrinsic kinase activity is going to phosphorylate the RNA polymerase. So once the RNA polymerase is going to be phosphorylated, it is going to have the lower affinity for the DNA or that particular region of the DNA, and that's how it's actually going to start the transcriptions. Then we have, so elongation is the steps in the elongation is going to be exactly the identical what we have discussed in the prokaryotes, where it's going to first dock onto the transcriptional site 1, and then it's going to look for the A or G sequences, and then it will go for the transcriptional site number 2, right? So that's those events are same, right? Then it will reach to the transcriptional terminations. So transcriptional terminations are going to be different in the case of different types of RNA polymerase. You can have the RNA polymerase 1, 2 or 3, and the transcriptional factors which is responsible for the termination of these transcriptions are going to be different. So RNA polymerase 2 transcription gene may continue up to the hundreds or even thousands of nucleotides beyond the end of a coding sequence. Then the cleavage of the RNA strands occurred by a complex which appear to be associated with the polymerase. Cleavage of RNA is coupled with the termination process occur at the same consensus sequence. The polyadenylation of the mature poly to messenger RNA occur at the 3' which results in a poly-A tail, which process is followed by the cleavage and terminations. Both the process, polyannihilation and termination, occur at the same consensus sequence, and both these processes are independent. So you see that if you have a polymerase A chain gene, it's going to be row dependent terminations. If it's a pol 3 genes, it's going to be row independent terminations, and if it is a pol 2 genes, it's going to be more complex, where you can have the pol 3 terminations generally coupled with the RNA processing event, in which the 3' end of the transcript undergoes cleavage and the polyanihilations. So transcriptional terminations, so it can be a poly-A dependent termination, which means it's going to form the polyanihilations at the end of the RNA, and that's how it's going to do the terminations. This type of terminations are basically coupled with the RNA maturing process, in which the 3' end of the nascent RNA undergoes polyannihilation and cleavage, and these 3' end processing reactions are carried out in 2 steps. In the step 1, the transcription of a poly-A followed by the cleavage of the nascent transcripts, and then the upstream product is polyannihilated and downstream product is degraded. Basically, 3' end processing starts when the cis acting element in the poly-A site of the nascent RNA transcript is recognized by the binding factor. So you can see that when it reaches to the stop codon, or when it reaches to the termination site, the RNA is going to be released from that site, and then it is actually going to do the polyannihilations with the help of the polyannihilation machinery. And the step 2, when these factor binds at the 3' end, it forms a very complex structure which results in a high shear forces, consequently processing slow down, which causes the disruption of the pol 2 and DNA RNA hybrid complex, and ultimately the termination occurs. So you can see that when the polymerase chain reactions or the RNA polymerase reaches to the termination site, it actually forms a very complex structures, and because of this complex structure, it results in the high shear force, which means it becomes very difficult for RNA polymerase to move further up, and because of that, the RNA polymerase as well as the RNA DNA duplex is getting broken down, and that's how ultimately it's end up in the termination of the transcriptions. So this is all about the transcription in the RNA in the eukaryotes. What we have discussed is that the transcription in the eukaryote is going to be very tightly regulated, because it depends on the different types of transcription factors. You might have seen that we have different types of transcription factors, the formation of the initiation complex is not possible until these transcription factors or these factors are not going to assemble onto the promoter site in a sequential manner, right? So because of that, they are actually going to be regulate the transcription within the eukaryotes. And apart from that, the termination in the eukaryotic transcription is also being more complicated than the termination in the prokaryotic system. So what we have discussed about the transcription in the eukaryotes, and we have also discussed about the transcription in the prokaryotes. Now once the transcription is done, then it is actually going to form the different types of RNA molecules, right? Now these RNA molecules, whether it is the ribosomal RNA molecules, whether it is the tRNA molecules, or whether the messenger RNA molecules, are susceptible, right? For example, the messenger RNA molecule is going to be a single stranded DNA, a single stranded RNA molecules, and these single stranded RNA molecules are actually going to be susceptible for the different types of RNases which are present within the cytosol, because once these RNA molecules are going to be synthesized within the nucleus, they are actually going to be transported outside into the cytosol, right? And once they are present into the cytosol, they are susceptible for the different types of damages. Like for example, the messenger RNA is going to be susceptible for the RNases, Same is true for the tRNA and same is true for the ribosomal RNA. Apart from that, some of these RNA molecules are going to be modified, right? Some of these molecules require the different types of modification. So these modification is required for the two purposes. One, it is actually going to provide the stability into the system, right? It's going to provide the stability into the molecule so that the messenger RNA or the ribosomal RNA or tRNA is going to be functional for a longer period of time within the cytosol, and also it's actually going to be used, like that modification is going to allow the functioning be more optimal. So it's going to make the optimal functioning of that particular molecule. So once the modification is being done, it's actually going to make the molecule more optimally catalyzing the transcription. So we are going to discuss about the post-transcriptional modification, because the RNA or the any RNA, whether it's the ribosomal RNA, tRNA or messenger RNA, when it's going to be formed, it is going to be modified so that it is actually going to achieve the stability and it's also going to achieve the optimal conformation so that it's going to participate into the downstream events like the translations. So when we are talking about the post-translational modifications, we have to discuss about the post-translational modifications of the messenger RNA. We have to discuss about the ribosomal RNA and the tRNA. So post-translational modification for the messenger RNA. So steps in the post-translational modifications, what you require the three different types of post-translational modification. You have to add a cap to the 5' end, you also had to add a poly A tail to the 3' end, and then you also required to splice out the introns. So let's first start discussing about how you can be able to add the cap to the 5' end. So the adding a cap to the 5' end. So this is the cap structure. What you see here is that this is the 5' end of the messenger RNA, and this is the structure. What you see here is actually a cap structure. So this cap is made up of a modified guanine molecule which is called as 7-methyl guanisine. Okay, and it is connected to the messenger RNA through the phosphodiester linkage. So the capping is in eukaryotic cell, the messenger RNA is inherently unstable at the end. So need to modify the end to protect it against the ribonucleases. Messenger RNA is capped so that it is protected from the ribonucleases, as well as it is important in binding the messenger RNA to the ribosome for the translations. It uses a certain cap binding protein complexes, right? So the capping reaction starts soon after the transcription has started. As soon as the 20 to 30 nucleotides are formed, the capping occurs, right? So at the 5' end, the capping process occurs. A slightly modified guanine, like the 7-methyl guanisine, is attached to the backward by a 5' to 5' linkage to the triophosphate of the first transcribed base. Capping reactions includes the condensation of the GTP with the triophosphate at the 5' end, followed by the methylation of the guanisine at the N7. Further methylation occurs at the 2-hydroxyl of the second and third nucleotide adjacent to the cap. So this is what you happen, right? You have the 5' nucleotides, right? So you are going to have the phosphohydrolase that is going to remove the gamma phosphate, right? And then it is going to form then you going to have the modified the guanine, right? So guanine is going to be also going to have, so this is going to be a GTP, right, where you also going to have the gamma phosphate, and then there will be an enzyme catalyzed reaction. So because of the guanyl transferase, this guanine is the phosphate guanine is going to be connected to the RNA molecules to form the cap, and then this cap is going to be methylated with the help of an enzyme which is called as a guanyl 7-methyl transferase, and then this methyl is going to be attached to the guanine molecules, and then you are going to have another round of methylation at the 2' positions, and that is going to be catalyzed by the another enzyme which is called as 2O methyl transferase, and that's how you are going to have the M7 guanine this cap, right? So this is going to be the cap which is going to be connected onto the messenger RNA. And this, what is the function of this capping? The capping is having the two function. One, it is actually going to provide the binding site for the ribosome site. Is going to provide the binding site with the cap binding proteins, and it's going to have the another function which is it's going to provide the stability. So this cap is not useless. If there's no cap present in the messenger RNA, that messenger RNA is not going to participate into the subsequent downstream translation events, because it's not going to have the efficient binding to the transcription to the ribosomes. Then the second is the addition of a poly-A tail, and this event is called as the tailing event. So the eukaryotic messenger RNA has a series of adenine residues ranging from the 80 to 250 in a number, forming a poly-A tail at the 3' end of the primary transcripts. This poly-A tail has several uses. For example, it can export the mature messenger RNA out of the nucleus. It increases the stability of the messenger RNA, and third, it serves as a recognition sequence for the binding of the translational factor during the initiation of the translations. The process requires the template independent RNA polymerase activity called catalyzed by the enzyme which is called as the poly-A polymerase. So what happens is that when the RNA polymerase reaches to the termination site, then we have the binding of the CPFC, and these CPFCs are actually going to bind to this consensus sequence, and then we have the binding of the CSTF and CF to the polyadenylation sites, and then it is actually going to allow the binding of the poly-A polymerase. And once the poly-A polymerase is going to bind to the messenger RNA, then it is actually going to catalyze the synthesis of the A RNA, and it is going to be non-template dependent. So it's going to be a non-template dependent synthesis of or addition of A residues onto the nascent polypeptide, nascent RNA chains, and that's how it is actually going to synthesize and add the varying amount of A. So this varying amount of A is actually going to decide the stability of this particular messenger RNA into the cytosol. It also decide the age of this particular messenger RNA. So if you have the 250, it's going to be more stable. If you have 80, it is going to be less stable. Apart from that, it also allows the export of the messenger RNA out of the nucleus. It increases the stability of the messenger RNA, and it serves as a recognition sequence for the binding of the translational factors. So that's why the whether it is a capping or tailing, both are actually responsible for the translation of the particular messenger RNA into the proteins. Then we have the third event. The third event is called as the intron splicing. So intron is a region of, so what will happen is in the eukaryotic genes, you have the genes, right? So you have a genes which are attached to each other, and that's why you are actually forming the genomic DNA. But in the genes, what you have is you have the some region of the gene which is not coding for the any kind of protein. So these non-coding sequences are called as introns, whereas these coding sequences are called as exons. So these coding sequence, these are the coding sequence, and these are the non-coding. These are non-coding sequence, right? And in an event when you want to do the messenger RNA synthesis, all these coding sequences has to be come together, which means if this is the gene, I have to synthesize, so when I will synthesize the messenger RNA, I will synthesize the messenger RNA from the gene, right? But from the gene, you're going to have the coding regions, you're going to have the non-coding regions. These non-coding regions, which are called as intron, has to be removed, and this step is called as the introns splicing. So introns are the non-coding nucleotide sequence which are present in a gene, and that do not code for the protein and don't appear in the final messenger RNA molecule, are removed by the splicing. Protein coding sequence of a gene, which is also called as exon, which are interrupted by the intron. So you in a particular gene you have the different regions, right? You can have the different types of regions. You can have the non-coding introns, you can have the coding introns, right? So these are the exons, these are the introns. And in if you want to make the protein, then all these exons has to come together, right? So all these exons has to come together, and that's how you can be able to have the proteins from this particular gene, right? So the vast majority of the eukaryotic genes are interrupted with the non-coding region intron, which needs to be spliced out, which means you have to remove these introns from the from the messenger RNA. However, histone protein coding gene is in the vertebrate is one among few exceptions. The occurrence of introns varies in the eukaryotic species. Some yeast species lacks introns, and many genes in eukaryotic carry a dozens of them. Few bacterial and archaeal genes also have the introns. So introns can vary in length from 50 to 20,000 nucleotides. In higher animals as humans, the introns are more than the exons. So there are four classes of the introns. You can have the group 1 introns, you can have group 2 introns. So both are the self-splicing introns and does not involve any protein machinery. Then you can have the spliceosomal introns. They are not self-splicing and you require a splicing machinery. And then you also have the introns that require ATP for the splicing. So there are four different groups of introns. One is intron, the group 1 and group 2 introns. These are the self splicing introns, which means they don't require any protein machinery. They can be just spliced out on their own. Whereas you have also required the spliceosomal introns. They are not self splicing and they also require a machinery. And then you also have the introns which requires the ATP for the splicing. So the splicing mechanism of group 1 and group 2 introns are the splicing mechanism of the both group 1 and group 2 introns involve the similar step of the true trans-esterification reaction, in which a ribose 2' or 3' hydroxyl groups makes a nucleophilic attack on the phosphorus, and the new phosphoroester bond is formed at the expense of the old, which means you can imagine that if this is a group 1 and group 2, then there will be an attack of the nucleophilic attack from the nucleotide what is present here onto the nucleotide attack onto this, and that's how this particular region is actually going to spliced out, and that's how it is actually going to form a linkage. So this means this OH is actually going to connect to this which is the phosphate group what is present here, and that's how it is actually going to form a phosphodiester linkage, and that's why this exon and this is the exon, right, got connected, and this is the splice the introns which is going to be removed. See, mechanism of these group transfer in nucleophile which is used. The group 1 uses the 3' hydroxyl group of the guanine nucleotide as a nucleophile. The group 1 introns are found in some nuclear, mitochondrial and chloroplast that code for the ribosomal RNA, messenger RNA and the tRNA. Whereas in the group 2, it is actually going to be formed by the lariat formations. And in the lariat formation also, whether it is a group 1 or group 2, the mechanism always the same, except that you are going to have the A at the nucleotide what is going to be utilized for the nucleophilic attacks, and then it's actually going to attack onto this, and then there is going to form a lariat, this lariat, and then this OH is going to attack onto the this chain, and that's how it's going to form the exon, right? So these two exons are going to be connected, and that's how it is actually going to release the intron in the form of lariat. Apart from this splicing mechanism, you can also have the alternate splicing mechanism. So alternate splicing means you can actually have the distinction between the different types of splicing event, and that's how you can actually have the alternate splicing in such a way that you can actually be able to have the different types of genes, for example, and that is very common in the case of the so alternate splicing is a method substantially used for many mammalian genes, can result in the multiple products that vary structurally and functionally from the same primary transcript. Sometimes the alternate splicing is upregulated phenomena, while in some it is strictly regulated. One of the best example of the regulated alternate splicing occurs in the sex determination of Drosophila. In Drosophila, three genes are involved in sex determinations: sex lethal genes or XL1, transformer genes, TRA genes, and the double sex genes. And these genes are getting the the going for the alternate splicing. So for example, in the alternate splicing, what will happen is that you are actually going to have the, so this is the gene, right, for that particular protein, right, and it's going to produce the primary RNA. Now in the primary RNA you have the exon 1, exon 2, exon 3 and exon 4. Now if these four exons can actually be able to do the alternate splicing in two ways. In one way, it is going to take the 1, 2 and 3. So if it takes the 1, 2 and 3, it is actually going to form the protein A, which means it's going to have the coding sequence what is present in the exon 1, exon 2 and exon 3. Now in the second example, it can actually take the exon 1, it can take the exon 3 and it can take the exon 4. So if that happens, and then it is actually going to form the protein B. So you can have the exon 1, exon 3 and exon 4, and that's why you see that the same gene, you have the only one gene, and that gene is actually providing the two different types of protein, the protein A or protein B, depending on whether it is going with the alternate splicing number 1 or it is going with the alternate splicing mechanism 2. And that's why the number of proteins can be very high, and that's why it's actually going to help in terms of the organism not to increase its genome size, but at the same time it actually can help in terms of producing the different types of proteins. You can even consider like it can actually go with like 1, go with the 3 and 4, right? That is what we said, right? It can be even like this also, 1 going with 2, then going with the 4. So alternate splicing is a very robust mechanism or robust tool what is present in the eukaryotic cell, and that is actually going to give you the different types of products from the single gene. Due to the alternate splicing, the functional genes are produced in female and non-functional genes are produced in the male. Alternate splicing occurs by the two mechanisms. One, by the two mechanisms. One, when two poly A or cleavage sites are available in the primary transcripts, the cleavage occurs at the one side, resulting in the two different products. Such mechanism is followed by the variable domain of the immunoglobulin heavy chain, and their diversity is due to the mechanism of the alternate splicing. Similarly, you can have the alternate splicing with such mechanism in the production of two different hormones. You can have the calcium regulating hormone in the rat thyroids and the calcitonin gene regulated peptide in the rat brain. The other mechanism involve the more than 3' splicing site for the 5' sliding site. Hence, the splicing occur in the taking either of these two results in the different products. So, we have a different types of examples where you can actually be able to use this alternate splicing. One of the classical example is the IgG, right? So IgG, you know that IgG has the two chain, right? One is the heavy chain and the other one is called as the light chain, right? So alternate splicing within these chains are, and then you also have the J chain, right, which is also called as junction chain. So IgG, you know that the different types of antibodies can be produced according to the different types of organisms and or different types of antigens, right? And all these antibodies are having the different types of structures, different types of sequences at their variable region, right? So if you see the antibody structure, right, you have this antibody, this is the antibody structure, right, where this is the heavy chain, right, this is the heavy chain and this is the light chain, right? But this region is actually called as the antigen binding region, and this region is very much variable, right? So from where this variation comes, you don't have those many number of genes. What happen is that these different combination of the genes and different combination of the exons, when they come together, they are actually being responsible for the formation of the different types of antibody molecules, and that's how they are actually being responsible for providing the diversity within the antibody structures. So because of the alternative splicing, you can be able to produce the different types of the antibody molecules. Similarly, you can have the alternative splicing, which can also be responsible for the production of two different types of hormones. One is calcium regulating hormone, which is going to be produced at the rat thyroid, and the other hormone, which is going to be calcitonin gene related peptide, which is going to be in the red base. So from the same gene, you can have the two different types of hormones. You can have the calcium regulating hormones in the thyroids, and you can also have the calcitonin gene related peptide in the case of brain. So hence the alternative splicing is very is a very robust tool what is present in the eukaryotic system, and that is also going to responsible for producing the different types of proteins. Then we'll talk about the processing of the ribosomal RNA. So processing of ribosomal RNA, eukaryotes have the 80S ribosomes, whereas the prokaryotes have the 70S ribosomes. Ribosomal RNAs are transcribed as a longer precursor sequence, which is then modified at the specific bases and cleaved to give you the mature product. In both bacteria as well as the eukaryotes, RNA processing involves the two basic steps, the cleavage and the base modifications. So RNA processing in the bacteria is different. So RNA processing in bacteria RNA precursor in bacteria is a 30S ribosomal RNA, which is modified and cleaved to keep the 23 ribosomal RNA, 16S ribosomal RNA, 5S ribosomal RNA, and some tRNA segment is between are also sometimes. So what you have is you have the pre-ribosomal RNA in the bacteria, and it's going to be a 16S ribosomal RNA that is going to be transcribed and that is going to be cleaved to give you the 16S ribosomal RNA, 23S ribosomal RNA, and 5S ribosomal RNA. 30S pre-ribosomal transcript consists of 16S ribosomal sequence followed by a spacer which may have the tRNA sequence in some cases, and then there is a 23 ribosomal sequence followed by the 5S ribosomal sequences near to the end point, 3' end. At times there is a one more tRNA sequence after the 5S ribosomal RNA sequence at the 3' end. There are 7 different genes for the ribosomal RNA in E. coli. They are essentially similar in the sequence of ribosomal segment but differ with the number and sequence of the tRNA segments. So the maturation process involves the methylation of the 30S ribosomal precursor. So this is the 30S ribosomal precursor what you have at the specific site occurring at the 2' hydroxyl group of the bases. Some bases such as uridine is modified to pseudouridine or dihydrouridine. Further cleavage process is carried out using the enzyme RNase 3, RNase P and RNase E at the site 1, 2 and 3 respectively. Intermediate products are formed, namely the 17S tRNA, 25S and 5S. They are acted on by certain nucleases to keep the final product of 16S tRNA, 23S and the 5S ribosomal RNA. So this is what you are going to have. You are going to have mature ribosomal RNA which is going to be 16S, 23S and 5S, and these will come assembled and give you the 70S ribosome. Similarly, you can have the ribosomal processing in the vertebrates or the eukaryotes. So in the eukaryotes, the nucleolus is the center of processing ribosomal RNA. At 45 precursor is formed by the RNA polymerase 1 and processed in the 90S peri-ribosomal nuclear complex to give the 18S, 28S and 5.8S ribosomal RNA. There is a tight coupling of the RNA processing with the ribosomal assembly. 5S ribosomal RNA is transcribed by the RNA polymerase 3 from a separate gene. So, in this case, you can have the pre-ribosomal RNA, which is going to have the 18S, 5.8S, 28S. So it's going to form a big transcripts, and then it is going to have the cleavage event. So this cleavage event is going to give rise to the 18S, 5.8S and 28S, and then you also going to have the 5S ribosomal RNA, which is going to be transcribed by the separately, right? And then the precursor RNA undergoes methylation at more than 100 bases from the 14,000 nucleotide at 2' hydroxyl groups. Furthermore, there is a modification of bases such as uridine to pseudouridine, etc., followed by a series of cleavage reactions. So cleavage and modifications are guided by the small nucleolar RNA. In each, the entire processing involves pre-ribosomal RNA, 170 non-ribosomal proteins, 70 SNRO RNAs and the 78 ribosomal proteins, and this is what is going to happen. So this pre-ribosomal RNA is going to form, then it's going to get the cleavage and it's going to form the 18S, 5.8S and 20S, and then there will be a separate synthesis of the 5-soluble amylase, and all these are going to assemble to give you the 80S ribosomes. Then we also can have the RNA processing in the tRNA. So in the case of tRNA, you can have the base modifications and you can also have the different types of modifications. So what is the processing in the tRNA? In both the eukaryotic and prokaryotic tRNA processing occurs. It is transcribed as a long precursor. Sometimes single primary transcripts carry more than one tRNA segment which are separated by the cleavage. Processing of pre-tRNA involves the cutting off of extra sequence by the endonucleases such as RNase P at the 5' end and RNase D at the 3' end. So in the tRNA molecules, what you have is you have the extra sequences. So in the extra sequences at the 5' end is going to be cleaved by the RNase P, and it's the extra sequences at the 3' end is going to be cleaved by the RNase D. RNase P is a ribozyme with the RNA exhibiting catalytic activity. After removal of the sequences from the 3' end, the CCA sequence is added by an enzyme tRNA nucleotidase transferase. So once the 3' end is going to be cleaved by the RNase D, then the 3' end is going to be added, like CCA end is going to be added onto the 3' end with the help of an enzyme which is called as tRNA nucleotide transferase. Now this end is over, right? The 3' end is over. This enzyme binds to the CCA sequence at its active site and phosphodiester bond is formed with the 3' end. Furthermore, there is a base modifications occurring simultaneously such as methylations, deaminations or reduction. In case of the pseudouridines, uracil is removed and attached to the sugar through C5. So after that there will be further cleavage and then there will be a base modification. So you can have the pseudouridine and all those kind of, and that's how ultimately you are going to have the final mature tRNA which is going to be formed, and this tRNA is going to participate into the translations. So this is all about the transcriptions, and what we have discussed so far. We have discussed about the transcription in the prokaryotes, and then we also discuss about the different steps, like we have discussed about the initiation, transcription, translation, and we have also discussed about the terminations. So you can have the intrinsic terminations or the row dependent terminations, and then we also discuss about the transcription in the eukaryotic cells or eukaryotic system, and where we have discussed about the composition of the RNA polymerase and different transcription factors which are responsible for the formation of the initiation complex. And then we also discuss about the promoter sequences and all of the kinds of machinery what is required for the transcription, and then we discuss about the post transcriptional modifications, where we discuss about the tailing as well as the capping in the case of messenger RNA. Then if we have discussed about the splicing of group 1, group 2 and all other kinds of introns, and then we also discuss about the relevance of the alternate splicing and how the alternate splicing is giving rise to the different types of proteins from the single gene. And then we also discuss about the RNA processing in the case of ribosomal RNA and as well as the tRNA. So with this, we would like to conclude our lecture here. In our subsequent lecture, we are going to discuss some more aspect related to the central dogma of molecular biology or the central dogma of life. So with this, I would like to conclude my lecture here. Thank you.